Today I'm going to talk about a different topic than I spoke about last week for the endocrine division, just in case there was any overlap in the audience. So both talks are about the role of the androgen receptor in breast cancer. And most of you have heard about the androgen receptor in the context of prostate cancer. But today I'll be telling you about what it does in a certain type of breast cancer called triple negative breast cancer. The talk last week, I think, was also recorded up in the endocrine division, and it was on the role of AR in the estrogen receptor positive setting of breast cancer. And of course, I wanted to address metastasis, and we'll get to that in this talk, because really that's the main problem now in breast cancer, because through surgery and adjuvant therapy, we can really cure most breast cancers, like up to 95% if they're still localized and non-metastatic. But metastatic disease is really the last frontier. If the tumor cells have already traveled to distant sites, and it's really hard to prevent progression of the disease and mortality. So that's really where we need to focus our energy these days. So I just wanted to first, at the beginning of the talk, before I might run out of time at the end, hopefully we'll have plenty of time for questions. But I wanted to show you some of the people in my lab currently that have done a lot of the work that I'll show you today. So Nicole Spoelstra has been in my lab doing excellent histology and all kinds of fancy multiplex immunohistochemistry of tumor infiltrating lymphocytes and hormone receptors in both experimental models in the lab, but also in serial biopsies that we get from clinical trials. I have 2 clinical trials as part of the grant that Tracy talked about with Dr. Elias, one of my clinical collaborators. And we get serial biopsies pre-treatment and post-treatment that we do analyses on in the laboratory. So Nicole's been with me for about 20 years now, and she's amazing. And a postdoctoral fellow in the lab right now is focusing on, I'll talk not so much about her work today, because she does a great job of talking about it herself, and she's really been the driver behind it. But she's looking at one mechanism that triple negative breast cancer uses to suppress the host immune system. And she had an F31 at Vanderbilt, an F32 in my lab from the NCI, and now she just got a great score on her K99R00. So I expect that she'll be getting wooed for her own faculty position and her own independent lab from many places soon. She's amazing. And Jessica Christensen in the lab started out as a postdoc from City of Hope, and now she's an instructor in my lab, and she has just obtained a Mediviver career development grant. And she's studying triple negative breast cancer to the lung, metastases to the lung, and how the lung cells there end up supporting the outgrowth of the breast cancer cells. And she also has a K22 that she's submitted. And then a graduate student who's about to finish up, Katie O'Neill, she sort of drugged me into metabolism, tumor metabolism, and the role of this cholesterol transporter in the lysosomes called MPC1, known for Niemann-Pick's disease, but we're looking at what it does in breast cancer. And it's higher in triple negative breast cancer than the estrogen receptor positive disease. And she also got this transitional award that is F99K00. And she's been being wooed for a postdoc position because she'll come with 3 years of funding, and just accepted a postdoctoral fellowship position at a great lab at UCSF. Liwei Ku, he's a cancer biology doctoral candidate as well, and just a second year, he'll be taking his computational exam soon. And he's studying also tumor metabolism, tryptophan catabolism in triple negative breast cancer, and how it helps the tumor cells survive, but also has paracrine effects and affects immune cells as well. And then Lindsay Crump actually just joined my lab last fall. She finished her doctorate in the cancer biology program in Tracy's lab. And she wanted to stay here in Denver for various reasons. And luckily for us, she decided to come do a postdoctoral fellowship in my lab. So she's studying immune suppressive factors that are regulated by the androgen receptor. And I'll show a little bit of that work today. But she also got a HERA ovarian cancer fellowship because some of triple negative breast cancer and serous ovarian cancer have a lot in common, actually, molecular level and the way they behave. So they make a lot of the same factors, and that's why Lindsay was able to get this fellowship. So a lot of great people in the lab, and of course, a lot of others that have left and gone on to do bigger and better things that have generated all the data that I'll show you today. And I just want to show all this because it just reminds me to say what a joy it is to work with all these really smart young people. And we were talking earlier about how it's great to take undergraduates for the summer programs, the Gates program and the others on campus, like the one from the R25 from the Cancer Center, because it's a chance for our trainees in our lab that are graduate students or postdoctoral fellows to get to mentor people themselves and really, pass on the favor. And since we don't have undergraduates on our campus, it's a chance for them to have them here for the summer. So we appreciate that. So let me tell you how I can't get into the whole story about how we got interested in the androgen receptor in breast cancer because it's quite a long story. And I told that one last week in the endocrine division. But I will say that it was a bench to bedside and back type of thing where we made this observation in a clinical trial that Dr. Elias was already running, that those that were resistant in estrogen receptor positive disease, the breast cancers that were not responding to traditional estrogen receptor targeting, had elevated androgen receptor. And that's actually how we got interested in it. And then we started doing a lot of preclinical modeling in my laboratory to figure out what was going on there, because as I said before, it's a known target for prostate cancer, but really very little was known about it in breast cancer. But normal breast epithelial cells do express the estrogen receptor, progesterone receptor, androgen receptor, and glucocorticoid receptors, the four main steroid hormone receptors. But the breast is really the most responsive to the female ovarian sex steroids, estrogen and progesterone. But one of the main therapies for estrogen receptor positive breast cancer is to take away that estrogen. And what we found is that they can actually start switching over to using the androgen receptor if you take away all their estrogen and start to act more like a prostate cancer cell would, and dependent on the androgen receptor. But what this slide shows here is what a group at the Dana-Farber in pathology there, they looked at the Nurses' Health Study, a whole bunch, like over 2,000 invasive breast cancers. And what they found was that the androgen receptor is even more widely expressed than the estrogen and progesterone receptors in breast cancer, because you find it in all of the three main clinical subtypes of breast cancer. So it's there in the ER/PR positive breast cancers that we say they're hormone receptor positive or luminal breast cancers. And those that we call luminal A versus luminal B, the luminal A have even higher than the luminal B, and then the HER2 positive that have amplification of the human epithelial growth factor receptor, they also have 68% of them express the androgen receptor. And then these triple negative breast cancers, they're defined as triple negative because they lack ER and PR and they don't express HER2. So, but what we found, and what this group here also published, is that many of these triple negatives have the androgen receptor. And the reason that was so important is now maybe we can target that, because triple negatives are usually only targeted with chemotherapy. And if they come back as a resistant disease and they're not responding to chemotherapy anymore, then we really did not have very many tools in our pocket to target these triple negative breast cancers. And now we have some immunotherapies like the checkpoint inhibitors and PD-L1 and PD-1 inhibitors, but there's really still very few targets for triple negative breast cancer, especially if they don't respond any longer and come back as metastatic disease that's resistant to chemotherapy. So today I won't have time to talk about, it's a whole nother talk, to talk about the androgen receptor in ER positive breast cancer or in the HER2 positive breast cancer. But I will say that a paper just came out showing that if you come in with an anti-androgen in patients that are resistant to trastuzumab in metastatic HER2 positive disease, they saw clinical efficacy of an anti-androgen. So that is just out if anybody wants to look at that. But today, what I'll really focus on is the role of the androgen receptor in the triple negative breast cancer setting. So the triple negative breast cancers, they represent about 10 to 15% of all breast cancers. The ER positive subtype is about 70%, so it's the most prevalent. But the reason we wanted to particularly focus on the triple negative is that it's has a peak risk of recurrence. If it's going to come back as metastatic disease, it does it really quickly. So its peak risk of recurrence as metastatic disease is within 1 to 3 years of diagnosis. So it comes back really fast. And they can also present quickly as interval cancers. And what they mean by that is in between the yearly mammograms. So it can grow quickly. And there's also a weak relationship between the size of the primary tumor and the lymph node status. So even small tumors can metastasize, and they can do it quickly. So in the median survival, if it's metastatic disease, is very low. So really, we want to focus on finding new therapies and new things that we can target in triple negative breast cancer, particularly after they become chemo resistant. So just to go through a little bit of the history behind this, about the same time as we started studying this, some other groups found that a subset, so these two papers in 2005 and 2006, found that there was a subset of triple negative breast cancers that expressed AR, as I showed you before, and they were androgen responsive when they modeled them in cell lines. And then this other paper from Brian Lehman, who's at Vanderbilt now, they discovered that there were actually multiple different types of triple negative breast cancer by doing RNA-seq, and they classified those that had high levels of the androgen receptor as luminal AR, because they actually, AR can, as I said before, kind of replace the estrogen receptor almost and make these tumors start having a little bit more of an epithelial molecular look about them, even though most triple negatives really look quite different. They lose a lot of their epithelial markers, and they start expressing a lot of mesenchymal and neuronal and strange and weird genes that epithelial cells do not typically normally express, so they become very dedifferentiated. The luminal androgen receptor high AR subtype is a little bit more epithelial, but it's still quite aggressive. And then the lowest subtype, the only triple negative subtype that didn't express much AR was the spatial-like subtype, but the HER2 enriched ones had, or the AR positive have a lot of HER2 in general, so that was kind of interesting. And then this last one also just broke the triple negatives down into these different subtypes, and really found that most of triple negatives, even if they don't have super high AR and they're not classified as luminal androgen receptor, which I think are, yeah, over here somewhere, a lot of them still have the androgen receptor. And you can kind of see that over here, like all these different subtypes, except for this one, express the androgen receptor. So this was the Lehman paper in 2011 that broke the triple negatives down further into these molecular subtypes. And they're listing some of the pathways that are high in the luminal androgen receptors. And indeed, some of these things like the tryptophan catabolism are things that we study and focus on now in the lab. But really back then, they did not know what is the androgen receptor doing and why is it super high on some of the triple negatives, but actually these also mostly expressed AR as well. And importantly, the androgen receptor is predominantly nuclear in the triple negative breast cancer. And I'll tell you more about the significance of that later, but the androgen receptor and the glucocorticoid receptor, they actually are not found in the nucleus unless there's enough ligand to drive them into the nucleus and keep them there. They, if there's no ligand around, they're in the cytoplasm. So this sort of nuclear localization really indicates that they are most likely active and because they're in the nucleus. So up here, we see several different types of breast cancer cell lines. And here I was also sort of putting the different cell lines that we commonly use into these different subtypes of triple negative breast cancer. But what you can see is almost all of the cell lines express some degree of the androgen receptor. And some of them are almost as high, like these MB 453s. They are almost as high as prostate cancer cells. And others express less androgen receptor, but what we found is if you try to knock it down completely, the cells die. So even those with very little androgen receptor are dependent on it for growth, and especially Anchorage independent growth. And I'll get into that quickly here in a minute. But I wanted to show you a little bit more clinical data here and why it's important to look at AR and triple negative breast cancer. So what the slide is showing is the probability of being alive. And this is after chemotherapy treatment, so neoadjuvant chemotherapy. About 20 to 30% of the triple negative tumors achieve the pathological clinical response rate, so like completely regress after chemotherapy. So in yellow, what you're seeing is the PathCR clinical response rate of the non-triple negatives. So these are doing pretty well. And then in blue, these are the triple negatives. So they achieved shrinkage of the primary tumor. And then they are doing fairly well, a little bit less well than the other subtypes that have ER and PR. But then if they had residual disease, so the RD stands for residual disease after the neoadjuvant chemotherapy, those that are non-triple negative are here in the gray dotted lines, but those that are triple negative and had residual disease, meaning the chemotherapy didn't make the tumor completely go away, are down here. So you can see that they have the worst outcomes, so the lowest probability of still being alive, say at 3 years, 68%. So subsequent studies showed that if you break the triple negatives down into these molecular subtypes, the luminal androgen receptor subtype with that high androgen receptor have one of the lowest pathological complete response rates. There's only one other type that has a lower response rate. So really, we want to try to hit the androgen receptor because those are the ones that are the most likely to come back and not respond to chemotherapy. And I'll show you more data here in a minute, but what we hypothesized is something that others from Kate Horowitz's lab had shown, that progesterone and the progesterone receptors influence stem cell-like cells in breast cancer in the ER positive setting. But in the triple negative setting, they don't have the estrogen receptor, they don't have progesterone receptor. So was the androgen receptor able to do the same thing in triple negative breast cancer as what Carol Sartorius here, and who also was in Kate Horowitz's lab as a postdoctoral, as a graduate student even, and later as a postdoc. But she and Peter Cabos had actually published some of this data showing that ER and PR were important to maintain the stem cell-like cells. but we were asking whether AR can do that in the triple negative. And there were some additional papers here that showed that in the ER positive setting, estrogen and progesterone receptors were important. So we started studying the androgen receptor in the triple negative breast cancer about the time that some new tools were coming out to antagonize the androgen receptor in prostate cancer. And I'm showing one of these right here. It was first called MDV-3100, but now it's called enzalutamide. And what you can see, these are some different types of antagonists for the androgen receptor. These are agonists. So here's testosterone that you've all heard about, high in males. But DHT, so women have adrenal androgens, in case you're wondering where these androgens are coming from in women compared to men. So women don't have as high testosterone as men, but they do have it. And these adrenal androgens can also activate the androgen receptor. So DHT, we like to use it as dihydrotestosterone because it's not aromatizable into estrogen, whereas testosterone can be converted to estrogen. So if you want to make sure you're studying androgenic effects, we use versus estrogen, we use DHT because it can't be converted to estrogen. But what you can see that was kind of interesting, Charles Sawyers, who's a really big wig in prostate cancer, he sort of tailored this enzalutamide compound by taking different characteristics of older anti-androgens and sort of modifying the chemical groups to take advantage of all of them and try to then screen for the one that would be the most powerful antagonist of the androgen receptor. And what was different about this drug that we decided to start using more of, because it was looking promising in prostate cancer and it was almost FDA approved in prostate cancer, and we decided to try it in breast cancer. And that's what we got the big Department of Defense grant on, that had 2 clinical trials in the estrogen receptor positive setting. We decided to use this one because it actually blocks the entry of the androgen receptor into the nucleus, whereas the old, so let me start here with the agonist. So if you have dihydrotestosterone around, it can get into the cell and bind to the androgen receptor, which is out in the cytoplasm until it binds to ligand. And then it's released from heat shock proteins and it dimerizes and can regulate different genes, a lot of genes. And that's why these ligand activated hormone receptors are so important, because they really control a lot of genes that are involved in growth and tumor progression. And they regulate things in the normal functioning tissues as well. So AR can then dimerize, it binds to coactivators, it turns on, it regulates a lot of genes. But if you have an antagonist around, this one, biclutamide, the older one, that's actually still used a lot in prostate cancer before they switched to the more expensive enzalutamide, this biclutamide actually still allows the androgen receptor to go to the nucleus and to dimerize, but it's in a configuration where it binds to co-repressors instead of co-activators. However, enzalutamide actually keeps it out of the nucleus, so it's a more complete blocker of its activity, because eventually what happens with biclutamide, similarly to tamoxifen, which is a competitive inhibitor of estrogen in the ER positive breast cancer, it can eventually start having agonist-like properties and acting like an agonist instead of an antagonist, and it can start then generating tumors that are resistant and are getting around this, and when it starts to turn into an agonist-like activity, whereas this has less, it has less of an opportunity to regulate genes still because it's not there in the nucleus. And then we used various other tools that antagonize AR in different ways. So this is some early data from our lab where we started using this enzalutamide, and we really did see that it kept in these breast cancer, triple negative breast cancer cells. If you add the agonist, dihydrotestosterone, the AR is in the nucleus versus the cytosol. It really goes nuclear, whereas it wasn't before. But if you add the enzalutamide now, you don't see it there in the nucleus. So that was really proof that it was acting the way it was supposed to act in breast cancer, similarly to how it acted in prostate cancer. So again, here's some staining. You can see that when it's there, when the androgen receptor is there in triple negatives, it's nuclear. And I'm going to cut to the chase and tell you that these two different androgens, both the biclutamide, the earlier competitive antagonist, and the enzalutamide, have now shown this enzalutamide was used in 2018, and it was largely because of our data in the laboratory, our preclinical data, that a group at Memorial Sloan Kettering, where they get more triple negatives than we do here, they did the first trial of enzalutamide in triple negative breast cancer. And they got a pretty good response rate for being metastatic disease. I think it was about a 33% clinical benefit rate at 24 weeks. And that is actually pretty good for metastatic breast cancer. And so they did the triple negative trials and published the results here in 2018. And we were allowed to do the trials in the ER positive setting. And I can talk more about that later. But so I'm sort of telling you the bottom line here. And the enzalutamide did a little bit better than the biclutamide. And that was earlier, before enzalutamide was developed. But I want to show you the basic science that went behind even allowing those clinical trials to happen. So one of the first experiments we did was in this triple negative breast cancer cell line. It's one of those luminal androgen receptor lines. It has a lot of androgen receptor, and the tumor grows in xenografts in mice in response to dihydrotestosterone. It gets bigger. And then we did 2 different doses of enzalutamide, the antagonist. And one of them was this higher dose, but we didn't start till the tumors were already growing. And then a lower dose, we started right at the beginning. So the mice got the agonist and the antagonist. And you can see that we got a reduction in tumor growth with both doses. And it works best if you start it right at the beginning. And again, we looked at the tumors in vivo to see, when we took the tumors out of the mice, to see how the androgen receptor looked. And you can see that it's increased when there's ligand, but it's decreased by both of the different doses of enzalutamide. And what we were seeing was that DHT was decreasing the amount of apoptosis, whereas the enzalutamide was making the levels of apoptosis go back up. And so the tumors were dying. And this is just a quantification of the levels of the androgen receptor. So you can see that it was decreased by the enzalutamide. So that was one of the seminal studies that they used to be able to go forward with the clinical trials. And we published this back in 2014. We also looked at some of these other subtypes that were non-luminal androgen receptor. So they were triple negative that had less of the androgen receptor than the ones that I showed you previously. So they model some of the other types of triple negatives that they still have AR, but it's not whopping levels. So again, we found that in xenografts with this cell line, the enzalutamide could decrease the viability as detected by luminescence in these luciferase containing cells. So you can see that there's a lot less tumor burden here than there was here. And you can see that here as well, that the tumors weren't growing with enzalutamide, whereas they were in the control. And so the anti-androgen gave increased necrosis, and the amount of apoptosis increased as well, and the androgen receptor was decreased. So one thing that I thought would be interesting to you guys is the enzalutamide, one of the genes, we did gene expression analysis on the tumors that I was showing you previously, and xenografts of a couple of different cell lines. And one of the genes of interest was amphoregulin, which is a ligand that for the epidermal growth factor receptor, EGFR. So we were interested in this because it was an AR regulated gene that was going down with enzalutamide. So it was decreased at both 24 and 48 hours. And we did staining for amphiregulin over here in the tumors from the mice. And you can see that the enzalutamide decreased the amount of amphiregulin. But why this was important, oh, we also saw that DHT increased it. So the agonist increased it, the antagonist decreased it. And why this was important was some of the people in the field were saying, okay, these anti-androgens will only work in triple negatives that have a lot of AR, but we showed that it was working in these other cell lines that had lower amounts of AR. And this is similar to actually to the, when you target the estrogen receptor in breast cancer, they now go down to 1% positivity of the estrogen receptor in the breast cancer of a woman to treat with anti-estrogens. And so we were asking, well, how can, even if you have a low amount of androgen receptor or estrogen receptor, how can it still make the whole tumor shrink, even if only like 1% of the cells in the tumor have the hormone receptor? And we thought, well, maybe it's these secreted ligands like amphiregulin that are affecting the adjacent tumor cells that might not have the receptor for the hormone, but do have receptors like EGFR for the growth factors. And so we went on to sort of prove that that was the amphiregulin binding to the EGFR in the adjacent cells. And I'll show you a model of this later. But was how one of the ways that you can affect a lot of the cells, even if only a few cells have the hormone receptor. But we started to look more at how AR might be supporting these stem-like cells within the tumor. And that's what this figure from a paper from a graduate student a few years back shows. And it was a cancer research paper. She looked at mammosphere formation because that's sort of our standard for stem cell-like behavior in this Anchorage independent growth in breast cancer. And so what she found is if she knocked down the androgen receptor, and you can see how that looks here. So she's pretty effectively knocking it down in 2 different triple negative breast cancer cell lines. If she knocked it down completely and tried to make stable cells, they wouldn't grow back up after you froze them down. So the tumors are really dependent on the androgen receptor. And if you knocked it down and then looked at the ALDH positive population, it was decreased with the AR knockdown in both of these cell lines. And also she did mammosphere formation, and she showed that if you knock down AR compared to the control, the non-targeting control, that she got less mammosphere formation, but also decreased size of the mammospheres. And then she did sort of the drug equivalent here, where she's using the anti-androgen enzalutamide instead of just knocking down the receptor, she's blocking the receptor activity. And again, got less mammosphere formation and smaller mammosphere size in these 2 triple negative cell lines. So that was our hypothesis, that maybe these AR positive cells were slower growing, and that's why they aren't hit as hard by the chemotherapy, because as you know, chemotherapy tends to hit the rapidly dividing cells, but it was these stem-like cells that can come back after treating with chemotherapy. If they're not killed by the chemo, they can recur and start making the tumor outgrow again. So we tested this in the mice, and we used a triple negative cell line that you see up here, and we grew it as xenografts in mice, and we did chemotherapy, specifically paclitaxel. And we either started the anti-androgen at the same time, so in the red is simultaneously, and the orange is after the paclitaxel, because women can't stay on chemotherapy forever, but some of these endocrine therapies women can stay on for up to 10 years. So we took them off the paclitaxel and then had either simultaneous treatment with the anti-androgen or after the chemotherapy. And you can see that both of these regimens actually decreased the tumor burden. These were luciferase containing xenografts. And you can see down here, this is the Cal per measurement, so the tumor volume, was reduced. But then what you see here is when we took them off the paclitaxel, and these tumors started to creep back up. So some of them started to increase again. Whereas these that got the simultaneous treatment, they were all still going down after the chemotherapy. They were not starting to creep back up. So this is actually the basis for a lot of the clinical trials that are going on now at various different places with triple negative, where they're starting chemotherapy plus the anti-androgen together if they're AR positive, and instead of waiting till they come back as chemo-resistant disease. So it was really nice to see that stuff from the lab translate into the clinic. And this is showing you kind of the conclusion of this part with our model. So I'm going to show you more about how the androgen receptor supports anchorage-independent survival. But what we're showing here is that the antagonist or knockdown of AR inhibited the stem light cell population and the mammosphere formation. And I didn't show you, but we did these tumor initiation experiments where you go down as far as you can and the number of tumor cells that you inject and see how many it takes to form a tumor. And we really found that the ones that have AR you can go down really low to very few cells and still get a tumor. So they're tumor initiating. And this is showing you how we thought that even the cells that don't have the androgen receptor, if the cells that have it and they're upregulating the amphoregulin in a transcriptional way, so the amphoregulin is then released and causes in a paracrine manner these cells that might be AR negative but have the epidermal growth factor receptor, the amphoregulin then binds to those and drives their growth. So it can act and have effects in a paracrine manner, and you don't need the androgen receptor to be in a lot of cells to still get an effect and get tumor shrinkage. And as I showed you, the enzalutamide synergizes with chemotherapy in the models of triple negative breast cancer, and especially if you give them simultaneously. And like I said, we really think this is because with the endocrine therapy, the anti-androgen, you're hitting the slowly growing population that is going to come back because it's not hit by the chemotherapy. Whereas if you treat simultaneously, you're also getting the bulk of the tumor that's rapidly proliferating to die as well. So then you get less tumors coming back when you take the mice off the chemotherapy. So then it was actually really interesting how we figured out that the androgen receptor was supporting growth in the Anchorage independent condition, like in those mammospheres or on soft agar. It was actually a completely separate project. We knew that triple negatives in general survive better in Anchorage independent conditions. So, these are carcinomas. They come from epithelial cells. Epithelial cells are supposed to be attached to a basement membrane. And normal epithelial cells, if you scrape them off that membrane and they're detached, they undergo apoptosis. But carcinoma cells, especially the really bad ones like the triple negative that are pretty dedifferentiated and lose some of their epithelial markers like ecadherin and other things, they can survive in the Anchorage independent condition on soft agar or on these polyhemo-coated plates where they can't adhere. They can survive almost as well as like a hematopoietic type of cancer that you grow in suspension culture. So they don't self-destruct when they're not attached to something. So in a completely different line of investigation, we were looking at what genes changed when we cultured the triple negative breast cancer cells in forced suspension culture, where they couldn't adhere to the plate. And we knew the triple negatives lived, but the ER positives in general die. So we wanted to see what genes they were upregulating or downregulating to persist in this Anchorage independent condition. And the two projects came together in a weird way, because when we put all these genes into a pathway analysis a type of program called Metacore that would predict upstream regulators of the genes, sure enough, AR came up as a really common regulator of a lot of these genes that were increasing in the suspension culture. So then these two projects came together and we started confirming a lot of things that are upregulated by the androgen receptor when the cells are in this Anchorage independent condition. We also looked to see if the androgen receptor itself was increasing, and you can see here in these 3 different cell lines that in the suspension culture, and this was after just 24 hours, the AR starts to increase. In fact, in some of the cells, you could barely detect it until you put them in suspension culture, and then it really increased quite a bit. Whereas the glucocorticoid receptor was not increasing. So we started saying, okay, if you block this, if you use the anti-androgen receptor enzalutamide and you grow the cells in soft agar, does it inhibit their growth? And sure enough, it did very effectively inhibit their growth on soft agar. And as I showed you, it went up at the protein level and at the RNA level. And here you see in one of the cell lines, the BT549s, you can see it come up in the suspended condition. That was this here, the same cell line, but here we're looking at immunohistochemistry. And then we did dual staining for the cleaved caspase, a sign that apoptosis is going on, and the androgen receptor. And what we found is the ones that were upregulating the cells on a cell-by-cell basis, the cells that are upregulating their androgen receptor are not undergoing apoptosis. And you really rarely, if ever, see dual positive cells. So that was telling us that the androgen receptor was protecting against apoptosis in this Anchorage independent condition. And what this mimics is actually metastasizing through the circulation, because they have to survive in Anchorage independent conditions to be able to metastasize. So this is kind of a summary of some of our lines of research that have to do with AR increase in the Anchorage independent condition. And I'm going to get into a few publications that we published during COVID over the last couple of years that I actually haven't really talked much about yet. So I'll just again show you that in this cell line, you can see how much AR goes up in suspension compared to the attached. And then here's an actual person's primary breast cancer compared to a lung metastases. And often the AR is quite a bit higher in the lung metastases than in the primary tumor. So we started to propose that, like I said, the AR was protecting against apoptosis when the cells escaped the primary tumor. So you can take the primary tumor out by surgery, but these ones that have already become disseminated, perhaps AR was helping them stay alive during dissemination, and also was it helping them at the metastatic site. And as I was saying before, Jessica Christensen in the lab is specifically looking at lung metastases and determining how the androgen receptor, both in the tumor cells and in some of the lung cells, these alveolar cells in the lung, how AR supports the outgrowth of the tumor at the metastatic sites. So about the same time as we were studying all this back in 2015, a paper from Devon Lawson, who worked with Xenografts and now has her own lab, she had an interesting paper using triple negative patient-derived xenografts. And she didn't confirm this at the protein level, but what she did is looked at she did single cell gene expression analysis on the primary tumor versus circulating tumor cells versus those that had just arrived at the metastatic site and were still small versus those that were really growing out at the metastatic site. And she saw both the AR and TGF-beta at the gene level were increasing in the circulating tumor cells from these triple negative PDXs. So that was pretty interesting and supported what we were starting to think here. But again, they didn't really follow up on it much at the protein level at all. So that's when Valerie Barton and Jessica in my lab, they started to do all of some of this work that I was showing you before and published that in Cancer Research, showing that it was protecting against apoptosis in the Anchorage independent condition. And then just this past year in 2021, during COVID, we got a lot of data written up that was looking at what AR was doing in the Anchorage independent condition. And I'm going to tell you about some of this work, and I may have to speed up here. We had a surgeon, a breast surgeon from Japan come and visit us, and he started right before COVID, and he got to leave like February, right before COVID hit. So he was here for, I think, 2 years, and he started looking at secreted factors that are regulated by the androgen receptor. And now Lindsay Crump is following up on that. And the really cool thing is that some of these secreted factors that are AR regulated suppress the immune system. And that, of course, allows the tumor to persist and avoid immune surveillance. And so that's turning out to be quite an interesting project. And then we also looked at combining anti-androgens with the new type of CDK4/6 inhibitors, so cell cycle inhibition, along with the androgen receptor targeting. And then lastly, we had a paper in endocrinology where we really showed that it was this feedback loop between the androgen receptor and the TGF-beta pathway that was helping to support the tumor cells in the Anchorage independent condition. And I think I have some slides on a little dabbling of data from those 3 papers here. So these are some of the factors that go up in response to the dihydrotestosterone. And this is a xenograft that came from Alana Wilm. It's a patient-derived xenograft called the HCI 009. And this is sort of the history of this patient and when the PDX was made from the patient. And we stained it for the androgen receptor, saw that it had a lot of AR, saw that it grew even better when you gave mice dihydrotestosterone. And so we wanted to look at some of the different genes by RNA-seq. What was this tumor making when it was growing in response to the androgen? And then we confirmed some of the genes at the protein level down here. And some of these are like PSA that's measured in prostate cancer, that increases with DHT, but also some other genes that only a little bit was known about at all in breast cancer, more about them in prostate cancer. So let's see, I'm gonna skip ahead a little bit. This is just showing more about the RNA-seq that we did on this patient-derived xenograft. And what this shows is that if we use a different anti-androgen called sebaterenol, we could decrease that same PDX. It was increased by the agonist DHT, and it was decreased its growth with the anti-androgen called sebaterenol. And again, that decreases the androgen receptor, and it's another drug that people are investigating. So what the MD from Japan, he really started looking at these different secreted factors. He kind of overlapped all the genes that were regulated by the androgen receptor with all known secreted genes, and started looking at some of them other than, this is PSA, KLK3 is another name for PSA. So we are really starting to look at these in detail, and it turns out that one called PIP, for prolactin inducible protein, it's also AR regulated, and we showed that down here. And then its receptor called ZAG is also going up with the androgen receptor. And so we think there's an interesting feedback loop here that supports the growth of the tumor between this ligand and this receptor. And this one is actually used in the clinic. My chairwoman had told me, did you know this AR regulated gene is used to decide whether a metastasis of unknown origin is actually a breast cancer? And we started studying it more in the lab, but they do already actually use it in the clinic. So let me skip to the chase because I wanted to show you this because it's a collaboration with Tracy, who introduced me. And it turns out that we're both interested in this chitinase-like 3 factor. And it comes up when you deprive ER positive cells of estrogen. And it also comes up in tamoxifen resistant ER positive cells. But in the triple negative, and ER positive, we find that it increases with androgens. And Tracy is interested in it because it's also regulated by a protein that she studies called semaphorin 7A. So we're collaborating on this, but of course, as happens often, a lot of other groups are starting to work on this, and they're starting to show that it is involved in metastasis of breast cancer and melanoma. And this paper recently came out by Jack Elias, I think last year or so, yeah, 2021, showing that this chitinase-like 3 combined with PD-L1 can really inhibit both melanoma and breast cancer metastases, but they don't really know the mechanism of what's going on so far. So we're starting to look into its androgen receptor regulation more. And I am going to have to skip over this stuff. This was all of our TGF-beta work showing that AR regulates a lot of TGF-beta ligands, receptors, and intermediate signaling pathways. And then in turn, those ligands come back and upregulate the androgen receptor. And if you inhibit TGF-beta with this Lilly compound, you don't get upregulation of the androgen receptor if you inhibit TGF-beta ligand and receptor activity. So that was an interesting feedback loop. And we also found that AR really helps the tumor cells survive because it helps, AR regulates a lot of antioxidants, and the reactive oxygen species increases when the cells are in Anchorage independent conditions, like when they're metastasizing. And so that's one way that AR is also helping the tumor cells survive. And we now have, we know that some of our models that are syngeneic, so they are, you can put them into mice and look at mice that have a competent immune system, have a lot of AR, and they metastasize and then metastasize in the lungs, they still retain AR. So now we can also see what AR and AR regulated genes are doing to the immune system. And actually a lot of immune cells express the androgen receptor as well. So this is the end of the talk. And as I was telling you, now this has reached the clinic. And this trial showed a 35% clinical benefit rate at 16 weeks, treating with the anti-androgen in metastatic triple negative breast cancer. So we were really happy. This is a whole bunch of other trials going on now. And then, as I said, ours are in ER positive, but these were all in triple negative breast cancer. And it's really showing some promise, at least on a subset. The other power of those secreted AR regulated things is maybe we can measure them in the blood, kind of like PSA is used in prostate cancer. So we can predict which tumors might respond best to anti-androgens, because that's kind of the next frontier. Is we're getting clinical benefit in some patients, but why do some people's tumors respond and some don't? So, and this is the trials in ER positive with Dr. Elias that I won't talk about today. And I will end, and hopefully we had just a few minutes for questions, but these are a bunch of people in the lab and former people in the lab. And of course, the patients in our clinical trials and the patient advocates as well. And then other collaborators here on campus, and then additional ones at other institutions Cecilia Cano, she and I are starting to look at AR and how it affects mitochondrial function. So I will end there and take any questions.